PhD research scholar in the Department of Zoology, University of Delhi. I am doing my PhD under the supervision of Professor Thakur. So today I am presenting a part of my PhD research work. The topic is entitled Dissecting the Direct and Indirect Interactions of STAT3 with Human Papillomaviruses and its implications in HPV transcriptional control in cervical carcinogenesis. Cervical cancer is the major cancer among the women in the developing country. It ranks 4th globally, and if you talk about the Indian population, it ranks 2nd. So, the causative agent for this cervical cancer is the persistent infection of human papillomaviruses. So, the human papillomaviruses are the double-stranded DNA viruses. If you look into the biology of this virus, there are two proteins, E6 and E7. Those are the oncogenic proteins. They cause the immunogenic transformations in the host microenvironment. So, the virus alone itself is not capable of doing this whole event. There are the involvement of certain host transcription factors. If you look into the biology of the genome structure of this virus, the LCR region in the viral genome is the interface wherein number of host transcription factors bind and cause the upregulation of further downstream genes, which is E6 and E7. So far, there are many transcription factors including those bind onto the LCR of this virus. However, the genomic location or binding of this STAT3 is not yet investigated. Why STAT3? STAT3 is oncogenic transcription factor that has been found upregulated in many immunogenic transformations, including cervical cancer. Previous reports from our laboratory have shown that with the progression of the disease, this cervical cancer, the level of this STAT3 transcription factor increases. So is the increase in viral load. So in the present investigation, what I have done, we have investigated the direct or indirect interactions of this virus with this transcription factor with the viral genome. Previous reports have talked so much, our previous reports have talked so much about this transcription factor, its role in cervical carcinogenesis. So from there we concluded that it is a master regulator that plays a role in HPV persistent infection, maintenance of its copy number, integration frequency, and transcription regulation. But there are certain key questions. Today I am addressing this one key question as to how this STAT3 is interacting with this HPV genome. Is it directly interacting with the HPV genome, or there are certain indirect interactions? So, to address the first objective, which is the direct interactions, we did certain in silico analysis, including the manual analysis of LCR region of HPV16 genome. Then, we extended our study to the experimental validation with the chip-based analysis. STAT3 recognizes a particular sequence, which is the TTA in the flanking regions, and in between there are 4 to 6 base pair sequences, variable base pair sequences. So, we looked into that consensus sequences and found that there are certain 4 base pairs, 5 base pairs, as well as 6 base pair sequence that might present in the HPV16 LCR region. Further, we did FIMO analysis, which is in silico analysis, and we found and there are 20 sites and 17 sites in HPV18, 28 in HPV16, and 17 in HPV16 sites motifs that could where the STAT3 can bind. However, these all in silico analysis need experimental validation. So for that we did chip-based analysis. We did chromatin immunoprecipitation. So we did chromatin immunoprecipitation with HPV16 for HPV16 LCR region. We designed the primer for HPV16 LCR region and we did the chip analysis. However, we failed to detect any STAT enrichment in HPV16 LCR region. There are certain previous studies that have also talked about STAT3 binding. They said that there is no direct binding of STAT3 on HPV18 genome. We looked into the HPV16 genome. They said there is no direct binding. So they also proposed there might be any indirect interaction of STAT3 with viral genome. So here we summarize our first objective as to there are multiple sites on the HPV16 and LCR region. Those were proposed to those were proposed for STAT3. However, the experimental validation lacked any STAT3 binding on HPV LCR region. The next question arises: does STAT3 interact indirectly with the HPV LCR? So for that, we again started with in silico analysis of different transcription factors that bind on the HPV LCR. And why different transcription factors? Because we have recently reviewed that there are a number of transcription factors that can bind on LCR region and also reported experimentally, but not STAT3. So we picked all those transcription factors and did this string analysis to find whether the STAT3 is directly interacting or physically interacting with those transcription factors. So we came up with the conclusion that there is CJUN and CFOS. Those were the two transcription factors. Those were strongly interacting with the STAT3. CFOS, CJUN made a very they are proven oncogenic transcription factors, and their role has been proposed in cervical cancer also. But here this physical interaction that we are investigating is not reported yet. So we looked into this physical interaction. We did co-immunoprecipitation analysis in order to find the physical interaction of the STAT3 with CFOS and CJUN, and we found that STAT3 was physically interacting with CFOS and CJUN not only in HPV16 positive cell lines but also in HeLa, which is HPV18 positive cell line. So reversibly we checked that CJUN was also physically interacting. So this was cross-checked basically. The co-immunoprecipitation confirmed that physical interaction of STAT3 with CFOS and CJUN. The other question arises, what if we alter the expression of this STAT3? Are these oncogenic transcription factors CFOS and CJUN also getting altered? And what about the localization? What about the expression of viral oncogenic protein that we are talking about, E6 and E7? So for that we did ICC immunocytochemistry, and we found on increasing the expression of STAT3 by the introduction of this IL-6, we found the increase in the expression of this CFOS and CJUN in HeLa and CHA cell lines. We also, the expression was also quantified, and we found that the increase in STAT3 was concordant with the increase in this CFOS and CJUN, and upon the inhibition of STAT3 with static, we found the expression of CFOS and CJUN was also downregulated. Not only CFOS and CJUN, I was talking about viral oncoproteins, so we also checked the expression of altering the expression of STAT3 in the alteration of HPV16 oncoproteins. So we checked the expression of HPV16 E6 and E7, and we found upon IL-6 introduction the expression of E6 and E7 was increased along with STAT3, and when the STAT3 was suppressed with static, the expression of E6 and E7 was also suppressed. The similar study was conducted in HeLa cell lines, and the results were the same. So from there we were thinking of looking into the biomarker approach. So what we did, we analyzed the large transcriptomic data set that included around 305 patients, and what we found on TCG database, the cancer genome atlas, and what we found was the expression of CJUN was differentially upregulated in HPV16 positive cell line, and as the Kaplan-Meier analysis indicated, the higher expression of this CJUN was associated with the poor survival. From here, we concluded that there is a direct interaction of STAT3 with the AP1 family proteins, those are CFOS and CJUN, and they are forming a physical complex. This complex might be interacting with this viral genome in order to alter the expression of this E6 and E7, and further this upregulation of E6 and E7 regulate the expression of these oncogenic transcription factors. Thank you so much.
Thank you. You were speaking the line, and now it's open for discussion. Any question in mind? I think this was there is a lot of questions. Students should ask questions.
So there are several isoforms of STAT. So although you mentioned why did you particularly select this STAT?
Sir, I have also investigated the expression of STAT1, STAT3 and STAT5. So my lab has been working on STAT3 already, but I was also keen to see the expression of other isoforms. But I came up with the conclusion STAT3 is the only, along with STAT5, but not STAT5 is not to that extent was significantly upregulated, but STAT3 was found significantly upregulated in this malignancy, which is cervical malignancy. Moreover, our laboratory has been working on this since a decade, so there are several reports from the lab stating the role of oncogenic
question for the stat file, whether the stat file having any interaction with the C force and
Sir, I have not presented it here. There was another objective wherein we investigated the complex, that is JAK-STAT signaling complex, targeting the 84 genes from that complex, JAK-STAT complex, in the cervical biopsy tissue samples. So what we did, we investigated the expression of those JAK-STAT signaling related genes. That was another objective I have not presented here. So from there also these genes, like another gene, there were 7 other genes also, I have not presented it here. So yes, but not other STAT1 or STAT2, because also there are other reports stating STAT1 is the inhibitor somewhere to this STAT3. They have antagonistic role in that too in cervical malignancy.
Okay, I think what part of your work you have presented here? Is it your sole work or you have taken from others also presentation?
This is my presentation. This is my PhD work. There are 2 objectives that I have presented. I have not presented another objective. These are actually this is unpublished data. So this is open for any discussion. I can get reviews from the public and the audience in order to improve my study.
Any possibility of start the interacting with other protein and getting it phosphorylated?
Making it phosphorylated. I have not read about the like physical interaction is helping phosphorylation of STAT3. I have not read any study like where a protein is interacting with STAT3 physically and helping it to
How
phosphorylate.
you design the experiment to study that?
So what else we can do is, we were thinking of extending this study with the activated STAT3. Like I have here investigated the role of total STAT3, its association with the C4 zone. I am thinking of extending it with phosphorylated STAT3. But if the activated STAT3, because ultimately there is the role of activated STAT3 that is binding to the genome of the LCR. So I was thinking of extending it with phosphorylated STAT3, repeating these experiments with phosphorylated STAT3.
Anything which can be done in site-directed?
Yes, sir. There was another objective including site-directed analysis, but since this is my 5th year and I had to, there are certain limitations also.
Okay, thank you. Thank you for the presentation. So the next presenter, this is Thakur. Here the next presenter.
Objective including site-directed analysis, but since this is my 5th year and I had to, there are certain limitations also.
Okay, thank you. Thank you for the presentation.